Welcome to this deep dive. Today's mission is pretty incredible. We are unpacking a groundbreaking paper from the journal Science Advances, and we're looking at a completely revolutionary approach to cancer immunotherapy.
Yeah, it really is a massive paradigm shift because, for the last decade, we've watched engineered immune cells actually cure certain blood cancers.
Right, like using CRT cells against B-cell leukemias.
Exactly. You take a patient's own T cells, reprogram them to hunt a target like CD19, and they just wipe out the disease. But, and this is the big mystery we're diving into today, when you point those exact same assassins at a solid tumor, they fail.
They just hit a total brick wall.
They really do. And for years, the assumption was that solid tumors are just too chaotic, that the targets are simply missing.
But this new research says we've been looking at that failure all wrong, right?
Yeah, completely wrong. The premise of this paper is that the targets aren't missing. The tumor is actually wearing basically an epigenetic invisibility cloak.
An invisibility cloak. I love that. And they didn't just figure out how the cloak works, they actually engineered a new biological tracker to see right through it.
They did. An ultra-sensitive receptor.
So to really understand this solution, for you listening, we first have to thoroughly unpack why standard CAR T cells, chimeric antigen receptor T cells, fail in solid tumors.
Right. And it starts with the target itself.
Like if you're sending a microscopic hit squad into the body, you have to give them a very specific mugshot to look for.
Exactly. And an ideal cancer target has to meet two incredibly strict criteria. First, it has to be pancellular.
Meaning it has to be on literally every single tumor cell.
Yes. If even a tiny fraction of the cancer cells lack that target, they'll survive the therapy, multiply, and the tumor just relapses.
Right. And the second criterion?
It has to be completely absent on your vital, healthy organs. Otherwise, your engineered immune system will attack the cancer, but, it'll also attack your heart or your lungs.
Which is obviously catastrophic. So what was the specific mugshot these researchers focused on?
A protein called CD70. Now, in a healthy body, CD70 is heavily restricted. You really only find it on certain activated immune cells.
Which is okay, right, because the immune system can just regenerate those cells later.
Right, exactly. But in oncology, CD70 is infamous. It's aberrantly expressed in a huge number of solid tumors. We're talking 70 to 80% of clear cell renal cell carcinomas.
That's the most common kidney cancer.
Yeah, and it also shows up in a vast majority of ovarian cancers and about 25% of pancreatic cancers.
Wow. So on paper, CD70 sounds like an absolute gold mine for a target. Why are standard CAR-T cells failing to cure these tumors?
Well, the medical community's diagnosis has always been this concept called antigen heterogeneity.
Antigen heterogeneity, meaning it's patchy.
Yeah, exactly. When you look at a biopsy of these solid tumors under standard diagnostic imaging, the CD70 expression looks dangerously inconsistent. Some cells have a lot of the protein, but many appear to have absolutely zero.
Okay, I want to picture how this actually plays out, because I know this study used patient-derived xenograft models, or PDXs, for their live testing.
Right, where they take actual treatment-resistant human tumors and grow them in mice.
Yeah. So how patchy was the CD70 in those specific models?
So they focused on two very distinct kidney cancer PDX models, K5 and K7.
Okay, let's start with K5.
K5 is a classic VHL-altered kidney tumor. And when you look at it, about 50% of the cells visibly express CD70.
50%. Okay, and K7?
K7 is a totally different beast. It's a highly aggressive NF2-altered sarcomatoid kidney tumor. It grows incredibly fast. And in K7, only about 18% of the cells show any visible CD70 expression.
Wow, only 18%. So the researchers ran the benchmark test. They engineered standard CD70 targeting CRT cells. And the study mentions they used widely used structural designs. Right?
Things like the 28s, BBs, and 1XX receptors, which dictate how the engineered cell activates.
Right. And they hit these K5 and K7 tumors with massive doses, like 10 million engineered cells per mouse. I assume the CRT cells just cleared out the visible targets and ignored the rest.
That is the tragic reality of antigen heterogeneity, yeah. The standard CRT cells killed the cells expressing high levels of CD70. The immunological synapse formed, the activation threshold was met, and they did their job on those specific cells.
But they failed against the tumor as a whole.
Completely failed. They barely slowed down K5, and they did literally nothing to stop the aggressive K7 tumor. The cells that appeared to lack CD70 just survived the assault and repopulated everything.
It's like, okay, think of it like using a lawnmower set to a 3-inch height. You push it over the lawn, and it perfectly cuts the tall weeds. Those are the cells shouting with CD70.
That's a great analogy.
But the short weeds, the CD70 negative cells, they just slip right under the spinning blade. They survive, drop seeds, and within a few months, your lawn is ruined all over again.
Exactly. The biological threshold of a standard synthetic CRR is simply set too high to catch the short weeds.
But, I mean, if I'm looking at that regrown K7 humor, and 82% of those cells are genuinely negative for this protein, why not just look for a completely different target? Why obsess over CD70?
Because the researchers basically refused to accept the premise that the cells were truly negative.
Wait, really?
Yeah. They hypothesized that solid tumor heterogeneity was an illusion, that it was just a byproduct of our diagnostic tools not being sensitive enough.
Okay, time for a quick mini recap for you listening to lock this in. So, bullet 1, CD70 works for blood cancers but fails for solid tumors. Bullet 2, CD70 is a great target but looks dangerously patchy or heterogeneous.
Right.
And bullet 3, standard CRT cells only kill the obvious targets and let the rest of the tumor escape. So here is a direct question for you. If you were the scientist looking at this failed experiment, would you throw the CD70 target in the trash, or would you take a closer look at those surviving negative cells?
Well, answering that question, the researchers obviously decided to look closer.
Yeah.
And they discovered those cells weren't negative at all.
Okay, but if standard lab imaging says the target is gone, how do you prove it's still there? You'd have to build, like, a way better magnifying glass.
Exactly. They turned to a technique called ISTED microscopy.
ISTED.
Yeah, stimulated emission depletion. It's a super resolution imaging technique that actually bypasses the natural diffraction limit of light.
Because normal microscopes can only zoom in so far before the light waves themselves just blur the image, right?
Right. But ISTED uses a specialized depletion laser to essentially turn off the glow of surrounding molecules. It leaves only this microscopic, ultra-sharp focal point. And the key, they did this imaging specifically in the far red spectrum.
I'm guessing the far red channel helps cut through biological noise?
You nailed it. Biological tissues naturally emit a faint glow under a microscope. It's called autofluorescence.
Ah, okay.
If you're looking for a really weak signal, that natural glow will completely drown it out. Moving to the far red channel eliminates that background interference.
Okay, so they have this super-powered far red microscope. But to prove the cells weren't negative, wouldn't they need a baseline, like an absolute biological zero to compare it to?
Yes, and that's where CRISPR comes in. They used CRISPR-Cas9 gene editing to literally cut the CD70 gene out of the DNA of a sample of tumor cells.
So those are the true knockouts, 0 CD70.
Exactly. Then they took the apparent negative cells from those K5 and K7 tumors, and they measured their mean fluorescence intensity against the true CRISPR knockouts.
The moment of truth. What happened?
The results were undeniable. The apparent negative cells actually had a statistically higher fluorescence than the true knockouts.
Wow. So the target was physically there on the cell membrane.
Yes. It wasn't negative, it was ultra-low.
Ultra-low, meaning every single cell in that tumor has the target. Some are just shouting, while others are whispering so quietly that standard medicine just declared them silent.
That's exactly what was happening.
But biologically, why would a tumor cell bother expending energy to keep just a microscopic trace of this protein? Why not just shut it off entirely?
Well, that question cuts to the absolute core of tumor biology and epigenetics.
Okay, let's get into the mechanics of that.
Right, so every cell in a tumor contains the exact same DNA instruction manual. Epigenetics is the regulatory system that dictates which pages of that manual are read and which are glued shut.
Without actually altering the DNA code itself?
Right, the code stays the same.
Okay, let's visualize this. Imagine the tumor's DNA is a massive encyclopedia. The CD70 gene is just one specific page of instructions in that book.
I like that. So in the CD70 high cells, that page is wide open. It's marked by an active epigenetic tag called H3K4AIM3.
H3K4ME3, the open tag.
Yeah. The cellular machinery easily docks onto that page, reads the instructions, and builds high volumes of the CD70 protein.
But in the ultralow cells, it's different. Imagine there is a rogue librarian roaming the archives.
Yes. The rogue librarian is an enzyme called EZH2. The researchers mapped this exact mechanism. EZH2 mediates epigenetic silencing by adding a repressive mark called H3K27E3 to the DNA.
Wait, let's unpack that term. H3K27E3.
Sure. It stands for the trimethylation of lysine 27 on histone H3.
Okay. Histones are the proteins that DNA spools around, right?
Exactly. When EZH2 adds those three methyl groups to that specific lysine, it causes the chromatin, the DNA structure, to condense really tightly.
So it's like EZH2 is a rogue librarian clamping heavy iron paper clips onto the CD70 pages.
That's a perfect way to picture it.
The DNA physically bunches up. The cell's machinery can barely wedge the book open, so it only manages to read a tiny fragment of the instructions.
Which results in that microscopic ultra-low trace of the CD70 protein. It is a literal biological cloaking device.
Wow. The epigenetic silencing actively hides the tumor from the immune system.
Exactly.
Okay, let's do another quick mini recap. Bullet 1. Solid tumors aren't truly heterogeneous. They're actually pseudo-heterogeneous.
Right.
Bullet 2. The CD70 target is pancellular, it's everywhere, but it's pushed to ultra-low levels by EZH2-mediated epigenetic silencing. So director question for you listening. Knowing the target is actually there on every single cell, just hidden, how would you design a biological magnifying glass powerful enough to see it?
Well, because the target is pancellular, the researchers realized we don't need a new target. We just need a hunter that isn't blind to ultra-low expression.
Because standard CARs are too blind.
Yeah. Standard CARs require hundreds, sometimes thousands of target molecules on a cell surface to form a strong enough connection to trigger a kill sequence. So they had to ditch the purely synthetic CAR-R architecture.
And enter the HIT receptor.
Through the HIT receptor.
The HLA-independent T cell receptor. How is the engineering of a HIT receptor fundamentally different from the standard CARs?
It's basically a masterful piece of hybrid engineering. So a standard CAR has an external synthetic claw to grab the target and an internal synthetic signaling tail to tell the T cell to activate.
Right, purely synthetic.
Yeah. But a HIT receptor keeps the external targeting claw of a KRR, so it can recognize targets like CD70 on the surface. But instead of a synthetic internal tail, it physically grafts that claw onto the natural T cell receptor, the TFR.
Wait, so it plugs directly into the T cell's original factory-installed hardware?
Exactly. It co-opts the highly sensitive natural CD3 signaling complex of the T cell.
The natural machinery.
Right. Nature spent millions of years evolving this complex to be exquisitely sensitive. A natural TCR uses things called immunoreceptor tyrosine-based activation motifs, or ITAMs.
ITAMs.
Yeah, they act as highly efficient signal amplifiers. A natural T cell can actually detect and trigger a full immune response off of a single molecule of a foreign antigen.
A single molecule?
Yeah, so by splicing the CAR claw onto the natural TCR complex, the HIT receptor achieves that same single molecule sensitivity.
Okay, I have to push back on this, because this feels like a glaring contradiction in the history of immunotherapy. If the natural CD3 complex is capable of single molecule sensitivity, why did scientists spend the last 20 years inventing purely synthetic CARs with synthetic tails? Why wasn't this hybrid the default from day one?
That is a great question, and it comes down to the brutal reality of the tumor microenvironment. It's a trade-off between sensitivity and stamina.
Stamina.
Yeah, when you use the natural TCR machinery, you lose the engineered costimulation that is built into modern CARs. Standard CARs have internal domains like CD28 or 41BB that act like an internal battery pack. They provide secondary signals that keep the T cell alive, multiplying, and fighting for months.
So a hypersensitive natural T cell will spot the ultra-low target, tap it, but then rapidly burn out and die of exhaustion before it clears the whole tumor.
Precisely. Without costimulation, the HIT cells would suffer from severe exhaustion. So to fix this, the researchers had to genetically graft two separate costimulatory molecules directly onto the surface of the HIT T cells.
And those were CD80 and 41BBL, right?
Yes, CD80 and 41BBL, right?
Yes, CD80 and 41BBL. So they literally strapped massive external battery packs to these sensitive hunters.
They gave them the stamina to survive. And the results of this combined engineering were just astonishing. When they infused these CD70 HIT T cells with the CD80 and 41BBL battery packs into the mice with those stubborn K5 and K7 tumors,
what happened?
they achieved complete eradication.
Wait, even the highly aggressive K7 tumor that only showed 18% visible CD70?
Completely wiped out. The HIT cells saw through the epigenetic cloak, found the ultralow cells, and killed them all.
That is incredible.
And they didn't stop at kidney cancer.
Yeah.
They tested them on patient-derived ovarian cancer models, OV4, and pancreatic cancer models, PDAC2.
And
complete eradication there, too.
unbelievable. But whenever we discuss engineering a biological weapon this exquisitely sensitive, my mind immediately goes to collateral damage.
Of course.
If this HIT cell can trigger off a whisper of a target, how do we know it isn't just hyperactive? Like, how did they prove it was specifically hunting ultralow CD70 and not just indiscriminately carpet bombing all the tissue?
They designed a really elegant proof of specificity experiment. They took a PDX tumor and physically mixed it.
Like a mosaic.
Yeah. They made it 75% normal ultralow tumor cells and 25% true CRISPR-Cas9 CD70 knockout cells.
Oh, wow. The ultimate test of precision.
Exactly. They put this mixed tumor into the mice and sent in the HIT cells. And the HIT cells meticulously hunted down and eradicated the 75% that had the ultralow target.
And the knockouts.
They left the 25% knockout cells completely untouched and alive.
So zero bystander effect.
Zero. They only kill when they verify the target.
Okay, mini recap number 3. Bullet 1, HIT receptors merge CAR targeting with natural CD3 sensitivity. Bullet 2, they require grafted CD80 and 4-1-BBL for stamina.
Right.
And bullet 3, they successfully eradicated seemingly resistant kidney, ovarian, and pancreatic tumors without a bystander effect. So listener, here is your question. We've built an incredibly sensitive biological weapon, but if it can detect even a whisper of CD70, what happens if healthy, vital organs are whispering too?
Right, because that brings up the ultimate fear in immunotherapy. On-target, off-tumor toxicity.
Killing the patient while trying to cure the cancer.
Yeah. If you make a T cell that triggers at the slightest hint of CD70, and a patient's healthy esophageal tissue expresses even an ultra-low amount, you cure the cancer, but the therapy destroys their throat.
And the terrifying part is, we already established that standard diagnostic tools like flow cytometry are entirely blind to ultra-low expression. We can't just run a standard lab test on a healthy liver and trust a negative result.
No, we absolutely can't trust protein-level diagnostics anymore. So to predict the safety of these HIT cells, the researchers had to look directly at the DNA's physical architecture across the human body.
And they used a technique called ATAC-seq for that, right?
Yes. ATAC-seq. This stands for Assay for Transposase Accessible Chromatin using sequencing.
Okay, let's unpack the mechanics of ATAC-seq because it's such a brilliant workaround.
It really is. It utilizes a hyperactive enzyme called the TN5 transposase. This enzyme physically inserts sequencing adapters into open, unwound regions of DNA. But if the DNA is tightly coiled like, when EZH2 puts those heavy paper clips on it, the enzyme just can't get in.
So it's like looking at the blueprints of every house in a city.
Oh, I like where this is going.
You aren't searching the physical houses for the stolen bank money. You're just looking at the blueprints, the ATAC-seq, to see if the door to the hidden safe is physically capable of being unlocked before you send the police in with hypersensitive metal detectors.
That is a fantastic way to explain it. It maps accessibility. So they ran ATAC-seq across 30 normal human tissues.
Wow. How many cell types is that?
It was 222 distinct cell types. They were looking for any tissue where the chromatin around the CD70 gene was open and accessible.
And what did the blueprint reveal?
In almost all healthy adult tissues, the CD70 gene is locked down tight. It's enclosed chromatin.
So the therapy is completely blind to those organs.
Exactly. It's only accessible in activated immune cells, which we knew about, and which the body safely replenishes. But surprisingly, it was also open in about 1 to 3% of normal esophageal epithelial cells.
The esophagus, I mean, that's a vital mucosal organ. If the blueprint says the door is open there, how do you thoroughly test that the HIT cells won't tear the patient's throat apart?
Right, because you obviously can't just put a human cell therapy into a regular lab mouse to test it.
And you certainly can't unethically test it on a human.
Right. So to test in vivo safety, they had to build a humanized model. The researchers utilized NBSGW mice.
NBSGW mice.
Yeah, they are severely immunodeficient mice, specially bred to support human cell grafting.
Okay.
They injected these mice with healthy human CD34 positive stem cells. And over a few months, these stem cells physically grew a functioning human immune system inside the mouse.
That is wild. So they created a living vessel with a human immune profile to run the ultimate stress test.
Exactly. They infused these humanized mice with the highly sensitive CD70 HIT T cells. And the safety results were vital.
Did it cause toxicity?
The HIT T cells did not cause any more myeloid or B-cell toxicity than the benchmark clinical CD19 sees.
The ones that are already safely used in hospitals globally today.
Yep, same safety profile.
But what about the esophageal tissue that ATAC-SEC flagged?
For that, they used an in vitro model. They exposed primary human esophageal epithelial cells directly to the HIT cells in a controlled lab dish.
And did it destroy the tissue?
The HIT cells did clear out that 1 to 3% of cells that expressed the target, but they stopped right there. The vast majority of the tissue remained completely intact, structurally sound, and healthy.
Wow. So it demonstrated a profound safety profile.
It really did.
Okay, final mini recap for the listeners. Bullet 1, ATAC-SEC maps where the CD70 gene is physically capable of being read.
Right.
Bullet 2, the therapy was proven safe in humanized NBSG-W stem cell models. And bullet 3, it doesn't destroy the normal immune system or esophageal tissue. So listener, here's a question for you. Think about the sheer volume of failed cancer treatments in medical history. How many of those targets weren't actually missing, but just epigenetically hidden from our early, clumsy tools?
I mean, it really forces the entire field of oncology to reevaluate decades of assumed knowledge. Solid tumor heterogeneity, this prevailing theory that cancer cells are just too chaotic to target uniformly, might largely be an illusion.
An illusion driven by the limitations of our own instruments.
Exactly. This paper proves that stealth targets are newly unlockable with sensitive receptors like HIT.
It's a massive paradigm shift. But before we wrap up, there is one final provocative thought I want to leave the listeners with. Something buried in the study that hints at the immediate future.
Oh, you're talking about the experiment with the rogue librarian, the EZH2 enzyme.
Yes, the enzyme placing the H3K27Me3 paper clips on the DNA to suppress the CD70 gene. What did they do with it?
The researchers took those K5 and K7 tumor cells in the lab and applied a pharmacological EZH2 inhibitor, basically a drug designed to block the enzyme.
And what happens when you block the rogue librarian?
It stripped the epigenetic paper clips away. The chromatin opened up, and the cancer cells were forced to unmask themselves. They started producing high levels of the CD70 protein again.
Oh, wow. I see where this is going. It's a choreographed 1-2 punch.
Exactly. The future of solid tumor treatment might not be a single superdrug, but a sequence. A patient takes an epigenetic drug, an EZH2 inhibitor, to systemically strip away the tumor's cloaking device.
Forcing the cancer to loudly and uniformly announce its presence.
Yes, and right at the moment of maximum vulnerability, when the tumor is glowing with CD70, you infuse the patient with the CD70 HIT T cells.
Unleashing an inescapable airstrike. You force the enemy out of the trenches, and then you send in the most sensitive hunters ever engineered.
It is a beautifully elegant strategy. It targets the cancer's very mechanism of survival.
I mean, we started this deep dive talking about how much we crave clarity in a diagnosis. For a long time, looking at solid tumors was just a murky, frustrating endeavor. We thought the targets just weren't there.
But it turns out the tumor was just whispering in the dark.
Yeah. And now, with STED microscopy, ATAC-SEC mapping, and the incredible engineering of the HIT receptor, we aren't just listening harder. We're handing the immune system a megaphone and a biological magnifying glass.
The targets we thought were lost to us are suddenly back on the table. The hunt for a solid tumor cure is absolutely back on.
It certainly is. So, to everyone listening, keep questioning the obvious, keep looking closer, and we'll catch you on the next deep dive.